Hold up your hands. Press the palms together. They match perfectly. Now put your right hand into a left glove. It doesn't fit. It never will. Same fingers, same bones, same everything, just flipped. Every molecule in your body has this problem. And in December 2024, 38 scientists published a paper asking the world to ban a bacterium that does not exist yet. Two of them are Nobel laureates. Some of them spent years building the thing they now want banned. That's the strange part. Not that scientists are scared. Scientists are scared of things all the time. The strange part is that the people who understand this best are the ones begging everyone to stop. So what did they see? Let's start with the glove. In 1848, Louis Pasteur was looking at crystals under a lens. Salt crystals from wine barrels. He noticed something nobody had noticed before. The crystals came in two shapes, mirror images of each other. He sorted them by hand with tweezers, one by one. That was the first time anyone saw that molecules have a handedness, a left version and a right version. Lord Kelvin later gave it a name, chirality, from the Greek word for hand. Here's what Pasteur could not have known. Life picked a side. Every protein in your body is built from left-handed amino acids. Every strand of your DNA twists with right-handed sugars. Every bacterium in your gut, every tree outside your window, every fish, every mushroom, every virus that has ever infected you, all of it, the same handedness. 4 billion years of life and not one exception. 19 of the 20 amino acids in your body are chiral. Only glycine is symmetrical. Everything else picked left, and nobody knows why. There are theories. Circularly polarized light in star-forming regions. Meteorites. The Murchison meteorite that fell in Australia in 1969 carried an excess of left-handed amino acids up to 18%. Maybe the choice came from space. Maybe it was random. A coin flip in a warm pond, amplified over billions of years until the other option was extinct. Either way, the coin landed, and every living thing has been playing by that result ever since. Which raises an obvious question. What happens if you flip the coin the other way? For most of history, that question was philosophy. You couldn't build the other option. Then chemistry got good. In 2016, a lab in China built a working mirror image DNA polymerase, an enzyme that copies genetic code, but backwards. 174 amino acids, all flipped. It worked. In 2022, the same group built a mirror image T7RNA polymerase. 883 amino acids assembled from 3 separate fragments. It transcribed mirror RNA. Then they used it to make the RNA components of a mirror ribosome, roughly 2/3 of the machine that builds proteins. 2/3. That's where we are. And this is the point where you might expect me to say, and then they finished it. They didn't. Nobody has. A complete mirror ribosome does not exist. A mirror cell does not exist. In fact, as of January this year, there is no known researcher on Earth still trying to build one. So why the panic? Because of what happens on the day it works. And that's where the answer gets much stranger than a broken glove. Think about what keeps you alive right now. Not food, not water, defense. Your immune system is a pattern recognition machine. It has receptors shaped to grip specific molecular structures on bacteria. Shaped. Physical shapes, like a lock waiting for a key. A mirror bacterium presents the mirror key. The lock doesn't turn. That's one layer. Here's the next. Your body digests invaders with enzymes. Proteases chop proteins apart. Lysozyme, sitting in your tears and saliva right now, cracks bacterial cell walls open. Those enzymes are also handed. They cut left-handed proteins. Facing a mirror bacterium, they slide off. John Glass at the J. Craig Venter Institute put it plainly. Such an organism would be essentially unassailable to those enzymes. Now, zoom out from your body. Because your immune system is not the main defense on this planet. Bacteriophages are viruses that hunt bacteria. There are more of them than every other biological entity combined, and every bacterial species on Earth is being hunted by them constantly. Jack Szostak, who won a Nobel Prize in 2009, said he is 100% certain that mirror bacteria would be resistant to all normal phages. The predators wouldn't work either. Protists that graze on bacteria in soil and water, they're built from the same handed machinery. So picture a bacterium with no immune response against it, no enzymes that digest it, no viruses that infect it, and no predators that eat it. Not weak defenses, absent ones. And here's where it stops being about bacteria and starts being about you. You are not separate from that defense system. You are made of it. Your body is not a fortress with an immune system inside it. Your body is the immune system, 4 billion years of it, written into the shape of every molecule you're made of. The reason you survived breakfast is that everything you ate had the same handedness as you, so your enzymes could take it apart. A mirror organism doesn't attack that system. It just doesn't register in it. You would be, to it, a warm pile of nutrients with the lights off, and it would be, to you, invisible. Now, is that actually the end of the world? This is where honest scientists start disagreeing loudly, and you should hear both sides. The case for the ban goes like this. Ruslan Medjitov, an immunologist at Yale, wrote that it's hard to overstate how severe these risks could be. That if mirror bacteria spread through infected animals and plants, much of the planet could be contaminated, and any exposure to contaminated dust or soil could be fatal. Szostak wrote that the result could be catastrophic, irreversible damage, perhaps far worse than any challenge we've previously encountered. Irreversible is the key word. You cannot recall a bacterium. Sebastian Ohm, one of the report's authors, said it would be in the environment basically forever, because there is no real way to get rid of it. Not a weapon you can disarm. Not a spill you can clean. The comparison they use is invasive species. Deepa Agash points out that even if mirror bacteria reproduce slowly, having no predators and no parasites drops their death rate far below that of native bacteria. That asymmetry is what makes invaders win. A single fungus spread by humans has been linked to the decline of at least 501 amphibian species and around 90 suspected extinctions. That fungus obeys all the normal rules of biology. A mirror organism obeys none of them. Now the other side, and it's stronger than you'd expect. Ting Zhu, the researcher at Westlake University, whose lab actually built those mirror enzymes, published a response in Nature in September 2025. His argument is not that the risk is fake, it's about timing. He wrote that the creation of a mirror image organism lies well beyond the reach of present-day science. And he made this comparison. It would have been premature to ban alternating current long before the electrification of cities, or to ban molecular cloning far in advance of recombinant insulin because of hypothetical dangers on the distant horizon. Andrew Ellington at UT Austin was blunter. He said banning this now is like banning the transistor because you're worried about cybercrime 30 years later. And there's real medicine at stake. Mirror image molecules resist your enzymes. That's the danger, but it's also the point. A drug your body can't chop up lasts longer. Etelcalcetide, approved in 2017, is built almost entirely from right-handed amino acids. More than 20 approved drugs contain at least one. Ban the field broadly, and you might kill the medicine to prevent the monster. Then George Church at Harvard raised the problem nobody has solved. A moratorium, he said, is a semi-voluntary thing. Something only the good guys sign up for. So you've basically handed it over to the bad guys. So where does that leave us? Here's what's actually confirmed. Chirality is real. Mirror enzymes exist and work. Mirror drugs are in pharmacies. Your enzymes genuinely cannot digest mirror proteins. Here's what's plausible, but unproven. That a mirror bacterium would evade immune systems broadly. That it would face no natural predators. That it could be built at all in 10 to 30 years. And here's what's speculative. Whether such an organism would even survive outside a lab. Whether it could compete with 4 billion years of optimized native bacteria. Whether the doomsday scenario is a forecast or a worst case that never arrives. Nobody knows. That's not a cop-out. That's the actual state of the science. And it's exactly why the argument is so loud. What we do know is this. In February 2025, 96 experts signed a statement at Asilomar. In July, UNESCO recommended a global moratorium. In March this year, a UN scientific advisory board called for red lines. And in January 2026, the doomsday clock moved to 85 seconds to midnight, listing mirror life among its reasons. A technology that does not exist on a clock built for ones that do. Kate Adamala at the University of Minnesota was building toward synthetic cells. She stopped. Her reason was simple. There is no benefit of mirror biology that couldn't be achieved other ways with normal biology, and that's not a risk she thinks we should be taking. Pasteur separated those crystals with tweezers because he wanted to understand what he was looking at. He had no idea that the shape he'd found was the reason anything on this planet can eat anything else. We're now the first generation with the tools to build the other side of that mirror, and the first generation that has to decide before we do it whether knowing is worth it. So, do you ban a technology before the danger is real? Or is that the only moment a ban can still work? Tell me in the comments, because right now serious people are on both sides of this and nobody has won the argument. If you want more questions that are too big for a normal day, subscribe. There are stranger ones than this.